For 40 years, Alzheimer's research has organized its massive intellectual capital around finding the single molecular cause of the disease. And for 40 years, clinical trials testing broad-spectrum monotherapies against these targets have failed with absolute consistency. The field has fractured into competing camps. One literature argues the disease is caused by amyloid plaques. Another points to the tau protein. Others isolate mitochondrial failure or activated microglia. Each camp draws its own cascade, placing its preferred molecule at the very top. Read as rivals, these models contradict each other. They cannot all be the primary cause. But if we treat these frameworks as observations of a moving front, the contradictions disappear. Every one of these hypotheses is correct about the pathology. They are simply describing different stages of the disease across a human lifespan. In 1907, the same year Alois Alzheimer described his famous plaques, Oskar Fischer examined the same pathology. Fischer recognized the lesions as part of a morphogenetic process radiating outward from a central point. He had an instinct for process. The century that followed his death ignored that instinct, obsessing instead over the specific substance of the lesions. A disease that takes half a century to complete cannot be understood by asking which of its molecules appeared first. The necessary question is temporal. We have to ask which specific process is load-bearing in which decade, on which substrate, and in which cell. This chart maps a human lifespan from the 20s to the 80s. Viewed this way, Alzheimer's reveals itself as a stereotyped structural failure spanning 5 decades. It moves through 3 distinct cell populations across entirely different regions of the brain. We are going to track this chronological front from its quiet origins in the brainstem all the way to terminal failure in the cortex. This mapping demonstrates why single-target trials often result in failure. They treat a substrate-shifting process as though its primary target were fixed. Alzheimer's operates as a substrate-shifting process occupying the better part of a human life. The pathology begins here, in the 3rd to 5th decades of life, and it does not start in the cortex. It begins in a tiny brainstem nucleus called the locus coeruleus, specifically in its noradrenergic neurons. The locus coeruleus neuron is metabolically extravagant. It acts as an autonomous pacemaker, firing constantly while sustaining an enormous, highly branched axonal tree. Because it operates so close to the absolute ceiling of its energy capacity, it has zero reserve left to spend on repairing mitochondrial damage. As the neuron ages, its quality control machinery breaks down at both ends. NAD+, the currency of cellular metabolism, is severely depleted. At the same time, amyloid beta proteins physically obstruct TOM40, the import channel that mitochondria use to pull in new repair proteins. Without those imported proteins, the mitochondrium cannot repair itself. It depolarizes and swells. This metabolic crisis represents the earliest erosion of the same TGF-beta homeostatic system that will later fail in the hippocampus and cortex. This specific metabolic failure produces pre-tangle tau inside the brainstem. This happens in individuals in their 20s and 30s, decades before a single plaque reaches the cortex, and long before any cognitive decline. During this phase, patients do not experience dementia. As these aminergic neurons slowly degrade, the only outward signs are prodromal symptoms: disrupted sleep, blunted arousal, and depressed mood. For up to 30 years, this remains a private, metabolic affair of 50,000 brainstem cells. The question is how a localized brainstem failure migrates upward to become a forebrain disease. The locus coeruleus projects massive axonal branches upward, bathing the forebrain in noradrenaline. This neurotransmitter acts as a standing brake, tonically suppressing the activation of immune cells called microglia in the hippocampus. When the brainstem neurons die, that brake is removed, raising the inflammatory set point of the entire region. Simultaneously, those exact same dying axons release a secondary cargo. The pre-tangled tau that accumulated in the brainstem is released into the synapses. The hippocampus internalizes these toxic seeds through a specific uptake pathway called LRP1, templating the misfolding of its own native proteins. The exact anatomical projection that regulates and protects the forebrain during health becomes the vector of its destruction. This dual pressure arrival, immune disinhibition combined with pathogenic tau seeds, pushes the disease into its next act. We enter phase 2, covering the 6th and 7th decades of life. The front of the disease has relocated to the hippocampus. Healthy microglia are maintained in a quiet, surveying state by a specific signaling program called TGF-beta SMAD. Under the chronic stress of the released noradrenergic brake and arriving tau seeds, that signaling dysregulates. The microglia exit their homeostatic state. Through a receptor called TREM2, the microglia pivot into post-homeostatic trajectories. They gorge on myelin and cellular debris until their cytoplasm gridlocks with lipid droplets, leaving them metabolically senescent and highly inflammatory. Researchers have long debated whether microglia in Alzheimer's are harmful because they attack synapses, or insufficient because they fail to protect them. At the cellular level, these are the exact same mechanical act. When a microglion digests a protective neural structure, it is simultaneously destroying a vital scaffold and withdrawing a necessary service. This brings us to the second bridge. The disease transitions from a localized immune response into an active demolition operation, targeting a physical structure called the perineuronal net. The lipid-laden microglia use their internal droplets to assemble NLRP3 inflammasomes. This triggers the cells to stop secreting basic cytokines and start pumping out matrix metalloproteinases, enzymes designed to digest tissue. An inflammatory cell physically converts into a digestive one. At the same time, nearby dying cells release redox-active iron into the tissue. The perineuronal net absorbs this iron, sparking Fenton chemistry. This reaction generates highly volatile hydroxyl radicals that aggressively shatter the rigid proteoglycan core of the net. Finally, the microglia deploy the complement system, painting the damaged matrix with C1q and C3 proteins. These act as molecular eat-me tags, marking the structure for rapid engulfment and removal. Through these three synchronized arms, enzymatic cleavage, iron catalysis, and immune tagging, the cellular immune response becomes a localized demolition. The bridgehead is secure, and the stage is set for terminal cortical collapse. In the 8th decade of life, the disease enters phase 3. The front advances outward from the hippocampus and engulfs the cerebral cortex. Every thread of this biological failure converges on a single target, the parvalbumin-positive fast-spiking interneuron. These specialized cells provide the inhibitory signaling that paces cortical firing and generates the high-frequency rhythms required for working memory. Because of the proteolytic turn we just tracked, these interneurons are completely stripped of their perineuronal nets. They lose the structural envelope that buffers their intense electrical activity. A parvalbumin neuron has immense metabolic demands. Denuded of its protective shield, the cell's own baseline activity poisons it. Unable to regulate its ion exchange, the exposed neuron succumbs to massive oxidative stress and excitotoxicity. When these interneurons die, the cortex loses its primary source of inhibitory tone. The critical balance between excitation and inhibition breaks down entirely, severely degrading the brain's gamma rhythms. Dementia arrives as a rapid, self-reinforcing circuit collapse, triggered only after the intrinsic structural buffers are digested. These phases represent 3 expressions of one collapsing homeostatic system, manifesting in 3 successive cell populations over time. Recognizing this systemic unity resolves a glaring paradox in clinical neurology. Postmortem data reveals a subset of cognitively intact individuals who carry massive burdens of amyloid and tau, burdens fully in the Alzheimer's range, yet display 0 signs of dementia. Their brains possess a unified architecture of cognitive resilience. Surviving the pathology requires the joint preservation of all 3 layers: homeostatic microglia, an intact perineuronal matrix, and competent parvalbumin synapses. All 3 are maintained by the same shared TGF-beta signaling node. If the phases were independent, you could buy resilience by blocking just one of them. Because they are 3 faces of a single system, preserving only one layer is insufficient to stop the sequence. This temporal dependency explains the relentless, expensive failure of Alzheimer's clinical trials over the last 4 decades. If the load-bearing lesion changes with every passing decade, a drug is only useful to a patient who is actively in that specific phase. Testing an anti-amyloid agent, a drug targeting a phase 1 metabolic substrate, on a symptomatic patient who has already reached phase 3 is a chronological mismatch. By the time dementia arrives, the rescue substrate is gone. Therapeutics must be phase-matched to the decade they are administered. We need pre-symptomatic metabolic support in the 30s, immune restoration in the 60s, and matrix preservation in the 80s. The restoration of the shared upstream TGS-beta signaling node acts as the single intervention, with a mechanistic claim to stabilize the system across the entire arc of failure. And the single most compact, precise biomarker to measure if that homeostatic restoration is actually succeeding is the physical integrity of the perineuronal net itself. Alzheimer's remains a 50-year process in time. The medical opportunity exists in the decades before diagnosis. We have merely been fighting the right battles in the wrong decades.